In this screencast on DNA, deoxyribonucleic acid, we're going to discuss the history of its discovery as the genetic information, as the genetic material for living things. We're going to look at how heritable information provides for the continuity of life. In other words, that DNA is the carrier of genetic information from one generation to the next. It is the molecular basis for heredity. And to understand that, we need to look at the history of its discovery and especially know the structure, become experts in the structure of the DNA molecule. The proof that DNA is the carrier of genetic information involved a number of important historical experiments, and science in general, as well as specifically in the discovery of DNA and its structure, is a collaborative study, and often actually competitive. And all of these scientists and groups of scientists contributed in their own way to the building of understanding the DNA molecule and its importance in heredity. Thomas Hunt Morgan, he was the scientist that worked with fruit flies and did a lot of work identifying chromosomes, and he identified the fact that chromosomes are very important and related to an organism's phenotype, or its expressed characteristics, so that phenotypes had to do with specific chromosomes. In identifying the fact that genes are actually on chromosomes was a big discovery, but they still didn't have the question answered as to whether it was the protein part of the chromosomes or the DNA part of the chromosomes. And originally, scientists thought that it was the proteins that were involved, because both of those substances are or make up the structure of a chromosome, and scientists knew that the vast complexity of proteins. They basically figured that it must be the proteins that carry the genetic information from generation to generation. But setting out to answer that question was the endeavor of, again, a large group of scientists. Avery, McCarty, and MacLeod came to the conclusion that their evidence showed that it was indeed DNA that was the first genetic material. Essentially, what these scientists did in looking for the transforming principle was to isolate and purify both the DNA and the protein separately from a strep pneumonia bacterium. Then they would look for which would transform non-pathogenic bacterium into the virulent form. And so when they isolated each one of those separately, and in two separate experimental setups, they injected first the protein into the bacteria, injected this into mice, and they injected the separate DNA into the bacteria. And what they found is that it was the DNA that transformed the bacteria into the virulent form and actually killed the mice in the experiment. That led them to say, it specifically led Avery to suggest that it was DNA that was indeed the genetic material. The next contribution to take a look at were from the scientists Martha Chase and Alfred Hershey, and they performed what is called the classic blender experiment. In their work, Hershey and Chase worked with a specific bacteriophage, which is a virus that infects bacteria. And the bacteriophage, there's a diagram here, a model of a bacteriophage. It has the DNA housed in a protein coat. And they grew the phage viruses in two separate media, and they labeled them radioactively. They used a sulfur radioactive compound to label the proteins, and they used a phosphorus radioactive compound to label the DNA. That way, with separate radioactive labels, they were able to identify down the road and follow these radioactive markers when they infected the bacteria with those bacteriophages. This slide just shows a summary of the classic blender experiment. Pause the screencast and take a look at the steps of the work that Hershey and Chase did in their classic blender experiment. And the results of their blender experiment was pretty significant. What they found is when the protein coat was labeled with the sulfur, the radioactive label, and they injected the bacteria, the radioactive label stayed in the medium. As opposed to the phosphorus label, the DNA was actually injected into the bacterium itself. This furthered the idea that it was the DNA that actually carried the genetic information from one generation to the next. So these scientists, through the Hershey and Chase, through their blender experiment, were credited with confirming that it was indeed DNA that was what they called the transforming factor. Another contribution to DNA technology, to the science of DNA as being the genetic material, was from a scientist named Chargaff, and his ideas, his results are known as Chargaff's rules. And although we don't yet know the specific structure of DNA, science did identify the composition, the smaller units that made up the large polymer of DNA. And we found that there were 4 bases, and those bases were not in equal quantity. In other words, it wasn't each wasn't showing up in a specific equal ratio. And for now, these 4 bases, as we'll refer to them, using the letter A, T, G, and C, what we found were these rules, that A always equaled T and C always equaled G. So in other words, their relative compositions were the same, or their relative ratios. And so whatever amount you found A, we also found T. Now we didn't know about the structure yet, but that added to the puzzle. This brings us to the contributions of Watson and Crick. And what they did is they developed what is called a double helix model of DNA. They, like science has so often done, their model was inspired by a lot of these discoveries that we've already talked about, including Chargaff's rules, and also looking at some of the X-ray crystallography data from scientists Franklin and Wilkins. Rosalind Franklin's contributions to the structure of DNA went really overlooked for a long period of time, and part of that could very well be to the nature of the times and the fact that she was a female in the profession of science. Maybe didn't get the due credit that she deserved, and actually didn't live long enough to receive that credit, but was posthumously awarded credit for her great contributions. And what she did is she essentially took an X-ray of this molecular structure. It's called X-ray crystallography. And with this technique, the locations of the atoms in a crystalline structure can be precisely mapped by looking at the image under an X-ray beam. And according to her contributions, Watson and Crick deduced that the only thing that could actually give this structure here, this cross structure at any view, must have been a helical structure. And the fact that it's a double helical structure is an important structural concept in understanding DNA and its role in genetics. So Watson and Crick finally in 1953 published a simple article, a one-page article in the science journal called Nature, which proposed a structure, a double helix structure for the genetic molecule, the complex polymer called DNA. In their description, they suggested that there were nucleotide bases that were on the inside of this double-sided ladder. It was a twisted ladder, but a double-sided ladder with a sugar and phosphate backbone on the outside. So these colorful little steps that are kind of broken here in this diagram, those represent the bases, and this blue ribbon around the outside is what we're talking about with the sugar and phosphate background. And remember Chargaff's rule that a purine was on one side of the strand and a pyrimidine was on the other. That would always give them the same width, would give the molecule the same width throughout its entire length. The relationship of DNA to chromosomes can be shown here that the DNA molecule, remember, when it's all wound up and organized, is what makes chromosomes. So in other words, chromosomes are just wound up long strands of this polymer, DNA. DNA had to accomplish some very important functions. It has to be able to store the genetic information, has to be able to be replicated precisely so that it can be passed on from generation to generation without having varying amounts of genetic material from one generation to the next. It's susceptible itself to mutation or changes, and what's really important is that it has to be able to be expressed as a finished product through the phenotype of the organism. So we'll build on and come back to all of this as we continue to look at the structure of DNA. Back to the original basic structure of the DNA polymer. The molecule itself, it's a huge long polymer, meaning that it's a bunch of repeating units that make up the large molecule. But each subunit is called a nucleotide, and each nucleotide consists of these three things: a phosphate, a sugar, specifically that sugar is deoxyribose, OSE tells you it's a sugar, and then one of four potentially different nitrogenous bases. Right now, we remember those as A, T, C, or G. It's the sugar and the phosphate that are always the same. They make up the backbone of the DNA molecule itself, the outside of that twisted ladder. The sugar phosphate, repeating units make up the backbone of the DNA molecule, and the bases make up these rungs. And remember that it will always be a purine that is across from a pyrimidine, and it will always be A's across from T and C's across from G's. Those four different nitrogenous bases that can make up the nucleotide are adenine and thymine, cytosine, and guanine. And we'll revisit this again and again and again. But adenine always bonds with thymine across from each other in a DNA molecule. Cytosine always bonds with guanine across from each other in a DNA molecule. We call this a base pair. Adenine and thymine are always together, cytosine and guanine always together. We refer to this as complementary base pairing. There is specific molecular structure that causes this, that they will only go together, kind of like pieces of a puzzle. They only fit together chemically one way. If you were ever given one side of a DNA molecule, because of the complementary base pairing rules, according to Chargaff's rules, you would always be able to give what is called the complementary side. A and T always go together. C and G always go together. One side of the DNA molecule acts as a template for determining the other side. We'll have continued practice with understanding the structure of DNA, but you should be able to pause here and answer these questions. So remember, one of the big important jobs that DNA had to do is that it had to be able to be copied. It had to be able to be replicated. If a cell is going to divide, remember, in mitosis or even meiosis, there has to be an accounting for the genetic material in each of the new daughter cells. Therefore, there has to be a synthesis phase. So the replication of DNA is really important. The base pairing rules suggest that each side is a template for the new strand. This is really important. This is called semi-conservative DNA replication. Scientists identified the semi-conservative method of replicating DNA, again by tagging parts of the molecule to see where they end up. And this shows you can take a look at the differences in the outcomes of the replicative molecule or models here, but science did identify the semi-conservative, where each side ends up as a template for the other in replicating its DNA. This is a process that we will look at in greater detail next. So remember that, like so many things in science, that scientific discoveries often come about because of the collaboration and often some competition. Remember that all these scientists, their livelihood was the science that they do. And the race to understand things often leads to new discoveries. That's the beauty of competitive science. After you become very familiar with the importance of the structure of DNA and become experts in DNA structure, we will move to the process of how replication occurs and how the musts of DNA structure are fulfilled through replication. But then further, we will see how through what is referred to as the central dogma of life, how we go from a molecule all the way to proteins and the expressions of traits. So heredity takes place by going from the genetic material on a molecular level to the proteins that determine phenotypes in organisms. Understanding the structure of the DNA polymer, as well as the history of its discovery, is crucial to understanding how it serves as the genetic material.